Hi guys, I'm Peter, and today we're going to talk about the spookiest part of the cell, the cytoskeleton. I'm pursuing a career in voice acting, so be sure to support me in voting for us in the video contest. Speaking of actin, this molecule is one of the three main components of the cellular cytoskeleton, along with intermediate filaments and microtubules. The cytoskeleton is a constantly reforming dynamic network formed by these three components. The job of the cytoskeleton is to provide support and structure to the cell, and provide tracks so that vesicles can travel to the right places and endocytose correctly. It does its job even better than a UMass PBTA. The focus of this video is specifically on actin. To carry out its cellular functions, the actin cytoskeleton is regulated by actin binding proteins, which orchestrate different functions, allowing the actin cytoskeleton to change its own physical properties. Because the cytoskeleton is so multifunctional, dynamic, and specific, it has been suggested that it may play a role in receptor signaling processes. Our research question is, how do the structural and dynamic properties of the cellular actin cytoskeleton enable its role, if any, in cellular signaling and transduction? To study this, we're going to look at an antigen-presenting cell and its interaction with the T cell. The left part of this figure shows a cross-section of the T cell. The black arrows indicate the flow of actin towards the center of the cell, which is created by a pressurized force. On the right, we see the immunological synapse, which we're going to call the IS. In this synapse, we see actin foci forming a sea of branched actin molecules flowing towards the center of the cell. The T cell receptors, TCRs, shown in blue, are anchored in the actin foci and are interacting with the peptide MHC, shown in magenta, of the antigen-presenting cell. The peptide MHC acts as a signaling molecule that activates the TCR and induces a signal transduction event in the T cells that leads to an immunological response. The flow of actin in the T cell increases stability of the synapse and allows for mechanosensation between the two cells. This is accomplished by the interaction between the LFA1 protein anchored in the T cell and the ICAM1 protein anchored in the APC. In order for this interaction to occur, the LFA1 must be in the high affinity state. This high affinity state is reached when the cytoplasmic tails of LFA1 are pulled apart from each other. The force that pulls these two tails apart is supplied by the flow of branched actin in the T cell. A clutch molecule, shown in yellow at the bottom of the left image, is on one of the cytoplasmic tails and creates a drag force in the presence of actin flow that pulls one tail away from the other tail, thus activating the LFA1 and allowing it to interact with ICAM1. The actin network within the IS can be thought of as a single mechanical unit in which perturbations that affect the mechanical properties of one area can propagate to surrounding areas and affect distant force-sensitive proteins. In this graph, the x-axis shows actin flow rate and the y-axis shows phosphotyrosine activity, which indicates TCR activation. We can see that TCR activation is strongly correlated with the force produced by actin flow. Force production through actin flow seems to help in peptide discrimination, which is an essential feature of adaptive immunity. Distinguishing between different peptides likely arises from catch-bomb behavior of TCR peptide MHC interactions. From this paper, we have learned that the cytoskeleton plays a significant role in the mechanical crosstalk within the IS between T cells and APC cells. This mechanical crosstalk mechanism has many gaps to fill. The actin cytoskeleton, in particular, may regulate the activity of mechanosensors involved in this crosstalk, and these mechanosensors should be identified and further explored in future work. Future work should also address how these mechanical signals are integrated via actin networks. Here's our works cited, and here's the contribution. Sorry for the bad audio, thanks.